Hello, everyone. Welcome to chapter 26, the urinary system. So, as we get started with this one, as always, I like to start things off with a quote. And this one was, "Let him who would enjoy a good future waste none of his present." So, I think that fits pretty well with the urinary system. Waste. Okay, so let's jump into this, begin to look at what we're going to be seeing now. If you're taking this summer, throughout the summer 2020 semester, then remember every chapter objective is tested on those standard multiple choice exams online through Honor Lock or the testing center. Really depends upon, I don't even know yet because I don't know what's going to be open at that point. So what we want to look at, and I like to use a mnemonic to help us understand the basic function of the urinary system, is it makes a wet bed. We're going to see that it helps control the body in homeostasis. It helps keeps us homeostatic. It controls blood's volume. Now, let's look at what a wet bed is and kind of help us understand how our urinary system helps us maintain a wet bed. So, a wet bed. Okay. And to help us understand how the urinary system helps us keep a wet bed, this is going to represent our blood. This will be entirety of your cardiovascular system, all the blood in your body. Let's just imagine that. And then let's imagine here I have a kidney, and this kidney is going to produce urine. Now, what we're basically going to be doing with a wet bed is we are going to be adjusting the blood's volume and concentrations to keep us in homeostatic range. So certain things in our blood, we know already through endocrine system, how it is that we control ion like calcium by one of the ways that we did that was excreting it by urine. We peed it out, urinated it out. We know that's something that we did. So we're going to start off with helps control blood's pH. So acid-base balance is the first role in a wet bed is acid-base balance. So we're going to control the pH of the blood. Now, this will be more in the next chapter where I get into that, but let me talk a little bit about how this actually happens. Now, to get started, I'm going to say, okay, we got a wet bed. A wet bed is going to be acid-base balance. So let's look at acid-base balance. Let me close my email account down real quick because it's going to ding every time I get one, and we get tons of emails right now. Sorry about that. Okay, so a wet bed, acid-base balance. Now, what does that actually entail? A little bit of. Now, first off, we have hydrogen ions that will be in the blood, and we also have HCO3 minus ions in the blood, bicarbonate ions in the blood. We should know this is how majority of carbon dioxide in the blood is transported in that version, in that variation there. So what we can do with this material is pull that out of the blood into the kidneys. Okay, so that's going to help either do that or the other way around. Okay, we can go vice versa with that. We can adjust the acid-base balance by adding or removing hydrogen ions or even bicarbonate ions from the blood to the urine, or ultimately from what's to become urine to the blood. And that's one thing that can be done. The other thing is going to be control water volume. Water volume. So now we've got a wet bed. So what is a wet bed? What is the W? Is water volume. So if we've got water volume, we have H2O in the blood, which can be removed or even put back in under the control of ADH, antidiuretic hormone. Remember, retains water. That's one of the things we can do there. The other thing that I want to discuss, so we got a wet bed, electrolyte levels are controlled. So there will be various electrolytes. So let's say that I have, I don't know what I just hit. Camera got disconnected. Uh, Gosh, there we go. Is it back? I must have hit something and loosened a cord. Trying to move around while I'm teaching, hooked to wires that are running everywhere is not easy. So let's say I had um, ions here. We have electrolytes, various electrolytes. We're going to talk about electrolytes, and we can add those or we can put them back. So we can move electrolytes in and out from blood to the kidneys, kidneys to the blood, pee it out, not pee it out, excrete it, not excrete it. Toxin removal excretes wastes. So again, if I had some waste products that would be found in blood, things that we want to get rid of, body can remove that. So a wet bed. So we've got toxins, and I'm going to write toxins above that. So we know that that's the T in wet bed. Now, one of the other things that's big is blood pressure control. Now, how do we control blood pressure? Is remember, we can use our kidneys to produce renin, renin, which controls blood pressure here. Renin was produced by the kidneys, juxtaglomerular cells, that we'll talk about later on, and that they are going to be used to activate angiotensinogen into angiotensin to help control blood pressure. That's important in the ACE inhibitor drugs. Then we're going to see that it makes EPO. It's involved in erythropoiesis. So let's jot that in there, that it makes EPO, erythropoiesis, stimulating hormone. It is produced there as well. And then lastly, it makes vitamin D3. So that is the participates in the synthesis of vitamin D3. And then also involved in glucogenesis, I want to mention here, called neoglucogenesis. I do believe that's a very important thing to discuss. So I'm going to just put vitamin D. D3 here is also used and synthesized with making us a wet bed. Okay, this helps makes us a wet bed. Okay. Now, a wet bed here that we produced, acid-base balance, water volume, electrolytes, toxin removal, blood pressure control, erythropoiesis, and vitamin D, also glucogenesis as well. Now, so here, our urinary system, we just want to basically understand the urinary system is produced by various organs. We have two kidneys, and there are a little asymmetrical in position due to the liver, due to the location of the aorta, due to a variety of things like that, the way it sits in the body. Which makes urine being produced by the two kidneys. Then the ureters, the tubes attached to those kidneys, go down and transport urine to a urinary bladder, which is this hollow muscular sac that stores it temporarily, and then it will squeeze down to release that urine. And then urine leaving the bladder out a urethra. Now, in males, urethra is very interesting because in males, urethra is part of two systems. In the male, it is part of urinary and reproductive, but in females, it's only part of the urinary system. As my anatomy professor put it, that the male said basically men have a wastewater treatment plant right in the middle of an amusement park. So because it's part of urinary and reproductive. Now, When we urinate, we call this urination or micturition. So the elimination of urine through the urethra. Foley catheter may be inserted and utilized. Don't remove those. It always bugs me in like zombie movies and stuff. It's like, how did Rick Grimes get out of the bed without tearing his internal organs out and destroying himself? You can't just remove a Foley catheter like that, or Cillian Murphy's character in 28 Days Later just pulls that thing out. I'm like, no way, man. If they don't, there's a very specific way to deflate a Foley. So, yeah, okay. Anyway, so as we said, it makes it a wet bed. And so homeostatically, we regulate our blood volume and blood pressure. Now, how do we kind of do that a little bit more? And I want to talk about this as a way of helping us kind of gather this, is that we regulate blood volume by releasing things like EPO and also our water volume and electrolytes. So one of the things we can do is, if we can urinate water out, we reduce the volume. If we retain water, we keep the volume up. You can get rid of electrolytes, and that is going to, now how does that actually regulate volume and pressure? Well, let me kind of show you what's going on here. This is a basic principle behind why that if you have high blood pressure, you go on low sodium diet. If this was your blood vessel here, if this was bloodstream, and inside this bloodstream, we had sodium cations and chloride anions from salt that we ate. Okay. Then what happens is these are ions. Now remember, water is polar. And if water is polar, it has a partial negative charge on one end and a partial positive charge on the other end, because water molecules, the oxygen atom is more electronegative and pulled all the electrons towards itself, leading with 4 electrons sticking out here. And if you had AMP1 with me, we would have drawn that out together. You would have seen these electron pairs sticking out, making this end very negative and this end very positive. And because of this, you have oppositely charged ions here. There is going to be the Cl minuses will attract the positive charges, and the Na pluses will attract the negative charges. Well, what does this actually mean? Well, these ions here are hydrophilic, okay? If they're hydrophilic, water who is polar will be attracted towards it through osmosis, and that will start to increase the water, because fluid, osmosis, it's a real hoot. Water always moves towards a greater solute. The water moves in, increasing the water volume, and that starts to push against the blood vessel wall, increasing blood pressure, okay? So if you increase salt concentrations or increase fluid concentrations, where if you increase salt, you will increase fluid, you increase blood pressure. And I hope that makes sense and that you can understand why it happens, and I can take that little bit of extra time and show you something to help you guys grasp that. We also know that erythropoietin will increase the number of red blood cells, which is going to increase the resistance of the blood vessel. You're going to have more red blood cells who are going to be rubbing against the blood vessel wall to slow down the blood flow, making increasing the blood pressure because it increased resistance, more stuff. Renin, as well, we know, is a kidney produced enzyme that goes on to activate angiotensinogen. Remember I said that renin is like a pair of scissors where it cuts a piece of angiotensinogen off to make it angiotensin 1. Now, also, we can take our plasma ions. We know that ions are in the plasma, and we can selectively determine which ones we want to remove. For example, we might pee out sodium, potassium, chloride, calcium, whatever. Pick an ion, pretty much you're going to pee those out. And then also, for example, I gave an example where calcium cation, Ca2 plus, is absorbed using calcitriol produced by the kidneys. We can also stabilize the pH because we can control the hydrogen ion and bicarbonate ions by either releasing it into the bloodstream or peeing it out. This is hugely important, by the way, for the next chapter. Now, we also can help conserve valuable nutrients. For example, the body can actually prevent urinating glucose out and even help make glucose if our body is low in it. If we have our low in amino acids, our body could preferentially retain it. Okay. Now, also, we help excrete harmful compounds. We help the body detoxify poisons. Now, the liver breaks it down into less toxic forms. Then our kidneys assist that by excreting those compounds, getting them out of the body. They're out of the body. Now, the thing about these materials is basically this. These are water soluble. So you can't get rid of it without it being in water. You have to put it in water to get rid of it. The goal of the kidney is to basically sacrifice just the right amount of water to get that stuff out. But see, you have to get it out in water, okay? Now, let's take a look at our kidney's anatomy. Now, gross anatomy of the kidney, you'll see there are three principal layers. A renal capsule, a renal cortex, and a renal medulla. The renal capsule is this superficial layer here, mostly collagenous, covers the outer surface of the kidney called the renal capsule. Sorry, I was pointing to the cortex of my mouse and didn't realize I can't do two things at once, apparently. Our renal capsule is this thin outer superficial layer, and then the renal cortex, which is superficial portion of the kidney inside, and then the renal medulla. So capsule, cortex, medulla. Medulla is deeper. Medulla, cortex, capsule. Okay. Now, inside the cortex, underneath the cortex, we have the medulla. And the medulla has these triangular shaped structures referred to as renal pyramids. The renal pyramids are very triangular in shape, and at the tip of each renal pyramid, you will find renal papilla. Hearken you to like fingertips, palpable fingertips, papilla. The renal columns are the bands of tissue located between each renal pyramid. And a renal lobe is when I take a renal pyramid, half a renal column on one side, half a renal column on the other side, overlying cortex, bam, that's a renal lobe. You have a renal pyramid, half of a renal column on one side, half a renal column on the other side, overlying renal cortex, bam, you have a renal lobe. The renal sinuses are any of the internal cavities inside the kidneys. And then also we will see coming from each papilla, renal papilla, is a minor calyx. The minor calyces come from every one of the the renal papilla, minor calyces. When two minor calyces or more unite together, they produce a major calyx. Here we have two, here we have two, here we have three unite together to make a major calyx. The minor calyces are transporting and collecting urine from what is called the collecting system. They go to major calyces. The major calyces then take all this urine to the funnel called the renal pelvis. The renal pelvis then takes us this funnel into the ureter, and the ureter goes to the bladder. Now, the place where you find the renal, where the renal pelvis is right here at a place where there will be blood vessels as well, called the renal, the hilum of the kidney, or the hilum. We saw hilum of the lymph node, hilum of the spleen, hilum of the lung. Now we have hilum of a kidney. Now, this thing here, this is something you are going to have to learn and know, and probably will have you guys at some point, excuse me, list. And what we're going to do here is we're going to get this now. I'm going to give you guys a mnemonic to help you kind of learn it. Now, this back to when I was a kid, Ren and Stimpy in a cage play very cool animations in reality. It's a really cool animation, you know, Ren and Stimpy. If you guys don't know Ren and Stimpy, that was back when I was a kid. Happy, happy, joy, joy, happy, happy, joy. Ren and Stimpy was the thing when I was a kid. That's kind of like the precursor to SpongeBob SquarePants and Rick and Morty and that kind of weirdness. Okay, it's kind of that weird, but yeah. Anyway, so it works for me, and Ren and Stimpy in a cage play very cool animations in reality. Okay, so let's help us understand good old Ren and Stimpy in a cage. Now, what we're going to do to help us understand it, I keep clicking the wrong thing every time. Okay, so what I want to do is up top here, I'm going to write glomerulus. That's a good word right there, glomerulus. Down here at the bottom, I'm going to write renal artery. And over here, I'm going to write renal vein. And what we're going to basically do is make a horseshoe. We're going to go from here to the glomerulus, glomerulus down to here. Okay? Now, Ren and Stimpy in a cage play very cool animations in reality. Ren, renal artery, Ren and. So, Ren and Stimpy. Now, renal artery, Ren and Stimpy. Segmental artery. Ren and Stimpy in interlobar artery. Ren and Stimpy in a arcuate artery. Ren and Stimpy in a cage. C, cortical radiate artery. Now, these are also called cortical radiates. We were talking about that they have an alternate name in a minute. There will be what's called interlobar arteries. Sometimes cortical radiates are called interlobular arteries. And be careful if I ever do that. Now, in my normal exams, I always have a question where I will have interlobular and interlobar as a choice. Just always warning. So Ren and Stimpy in a cage, CA. Now, what we're doing, and Mr. Lever didn't give himself enough room here, is cortical radiate, CAGE. So CA is going to be afferent arterial. Now, what is afferent arterial going for? So remember, Ren and Stimpy in a cage, CAG, glomerulus. E, efferent arterial. So this means now afferent arterial arrives the glomerulus, efferent arterial exits glomerulus. So Ren and Stimpy in a cage. Okay, now play. They play. What do they play? Peritubular capillaries. And I'm going to go ahead and just change the blue here. Peritubular capillaries. Ren and Stimpy in a cage. P, play. very, venules, very cool, cortical, radiate, veins, very cool animations, almost arcuate veins, in interlobar veins, reality, renal veins. So, Ren and Stimpy in a cage, CAGE, play very cool animations in reality. Okay? And that is a useful mnemonic to help you remember the flow through this. This is something you do see. I had a student contact me. They were taking their TEAS test and they had this was on the TEAS. Um, I'm going to ask you guys questions like, where do you go next? That kind of stuff. Okay. Now, what we're going to do is so Ren and Stimpy in a cage play very cool animations in reality. So if you actually were to go Ren and Stimpy in a oops, sorry. Ren and Stimpy in a cage play very cool animations in reality. There we go. Bam. All right, that'll help us a little bit there. I always like to kind of do something like that every now and again when I can to help you kind of visualize more what the mnemonic is helping you try to remember. And mnemonics work. They help stick. I mean, it's so we want to help keep that in your mind, help you guys remember it and retain it for the long haul. Okay, so now, and this is another way of drawing it. I do mine the way I do my horseshoe. They do theirs. It really doesn't matter so much. I'm just keeping to the color scheme of the book as much as possible. Now, segmental veins do exist, by the way. It's just that our book doesn't seem to think, but they do exist. I asked my mother's nephrologist, and they do. I looked it up multiple times and asked a nephrologist. So now, next drawing that we're going to do is a big one. This is going to be for your lab's replacement points. So for lab exam 4, there will be a drop box to submit this, and we're going to draw what's called the nephron. Now, there are 3 distinctive regions of this, and we're going to start out by drawing it together, the 3 distinctive regions of the nephron. Now, what all you have to do is for lab is in the drop box is to put this into a drop box called the nephron. You can draw it on paper, snapshot it with your phone. Just go and take your phone, snapshot it. And then when you take that snapshot, you can use your phone and you can literally go in, log into D2L with your phone. You actually go to assignments when you see the replacement points for the lab. And now this is in our lecture lab combination page. We only have the one page. You'll then go in and distinctly do that. So it'd be kind of interesting, I think. I think it'll be kind of a neat way of looking at things, helping you. You can upload it into that. It's going to help you guys in lecture and in lab. Now, right now, I was just told that the departmental final for the spring 2020, that they have not gotten they can't get the webcams they ordered so everyone would have a webcam. Literally, we can't get them because they are in China and there's still restrictions on that kind of stuff. So we can't get them. So that means nobody would be able to have one. If they can't guarantee everybody has one, they can't proctor that exam. If they can't proctor that exam, I don't know what they're going to do with 2 weeks kind of at the, we're 2 weeks to the end of material. So, it's okay. So let's take a look at the nephron. Now, what I want to get started with is there's three distinctive regions. First one, renal, corpuscle, renal tubule, and then collecting system. Okay. And we're going to start all by writing those three anatomical regions. Now, in the renal corpuscle, let's look at what it's made up of. Now, when I draw it, what I like to do is draw it kind of like this. I've got my anatomical reasons to draw it this way. You do not have to draw it identical to me. Maybe you got a professor that you've taken before who drew it in a way that is better for you. As long as you include everything, I'm fine with that. Maybe you had Dr. Lockhart once before and he draws it in a way you like better, or Mrs. Glass draws it in a way you like. I think that's the only three of us who draw the nephron that I know of for sure. So this is the way I draw it, and I start off with this thing. Then what I do is I come around this, so the little, looks like a snake trying to eat this. And I'm going to kind of draw a line there, just so we know, kind of a dotted line, just so you know kind of what's going on. And then I'm going to make my snake kind of slithery. Then we're going to come down like this. Let's taper him off here. And then let's loop him around here. Then let's make him get thicker up here. And if you've had me for A&P1, then you're used to me dotting things like this. And what we're just going to do is this and this and that. Okay. So basically, this is our drawing right here. Okay, this is what we're basically drawing. Now we got it drawn, let's label it. So first off, I'm going to draw an arrow coming in and an arrow going out, signifying that we have blood entering and leaving. And this is the afferent arterial. Now remember, Ren and Stimpy in a cage, afferent arterioles. An arteriole is a small diameter. What did I just hit? Must have froze something here. I don't know where my computer froze up. What's going on? Hold on one second. I can't even tell what's happened here. I must have hit something that I didn't know about, moving around, and I want to be live on this, and it's not. I don't want that. I want the live feed. And, well, wonderful, wonderful one. Okay, something happened. I must have loosened something when I was going, and I can't draw now. I don't know what's happened. Gosh. And if I stop it, it just won't work. It just, if I stop it, I can't bring it back up. I had to stop recording. Okay, I'm going to have to stop recording and see if I can make this come back up.